Just to review here, we are in domain Eukarya, supergroup Archaeplastidia, and kingdom Plantae. Here's the taxonomy that we cover in this PowerPoint. It's not a lot, but I want y'all to get this taxonomy down now, because like always, we will continue to add to it as we go through Plantae. We start with clade Lycophyta. These are the ground pines or spike mosses. Their common names should make some sense based on their appearance. Remember, the sporophyte is the dominant generation here. Gametophytes are short-lived. The lycophytes were the first to evolve true roots, leaves, stems, and microphyls. We'll see a microphyll on the next slide. They have fertile leaves and a strobilus, which is just a fancy word for a spore-bearing structure. We will see them throughout this group. There are two genera in this phylum, Lycopodium, your ground pines, and Selaginella, your spike moss. You should be able to tell them apart both microscopically and macroscopically for lab. Who knows, maybe they'll show up in lecture too. Microphylls are literally a small leaf, but they are not always small. It's really just an uncomplicated leaf with only one vascular bundle in the center. This is Lycopodium, the ground pines. They are also sometimes referred to as the club mosses, which is a horrible common name because they obviously are not a moss, which we looked at in the bryophytes. Lycos means wolf in Latin, and podium means foot. To the right is a skeleton of a wolf paw. Let's flip it and rotate it. You can really see here where the name Lycopodium comes from. Here's a close-up of the strobili, the spore-producing structure on the sporophyte generation. This is your diploid generation. This is a cross-section of the strobili. You can see the sporangium, which is protected by the sporophyll. Inside the sporangium are the spore mother cells. This structure is homosporous, meaning it only contains one type of spores, which will develop into the gametophytes that produce both male and female reproductive parts. This is Selaginella, the spike mosses. Again, this is a horrible common name because they are not a true moss, which we looked at in the bryophytes. Here's a good example of a plant coping with its environment. Resurrection plant, a species of Selaginella, lives in Arizona and Texas, where you would expect to see nothing but desert cacti and brush. So what is a fern ally doing here? Well, they essentially die off, basically folding inward to conserve water during the droughts, which is what you see on the left. Once it rains, they reanimate, as seen on the right in this picture. This is a unique adaptation which allows this fern to survive even though it only has water available periodically. Here's a close-up of the strobili, the spore-producing structure on the sporophyte generation. This is the cross-section of the strobili. Selaginella has two types of spores. The microsporangium contains microspore mother cells which produce male gametophytes, while the megasporangium contains megaspore mother cells which produce female gametophytes. Here is a comparison between the strobili of the two genera. You will need to tell them apart for lab and perhaps for lecture as well. Remember, Lycopodium is homosporous. One spore type produces gametophytes containing both male and female reproductive parts, while Selaginella has two spore types. Megaspores produce female gametophytes, and microspores produce male gametophytes. This is phylum Monolithophyta, which contains your true ferns, Celotum and Equisetum, which are two fern allies. Ferns have a photosynthetic gametophyte that consists of a prothallus, a photosynthetic leaf-like structure, not a true leaf, and both male and female reproductive organs. The leaves of a fern, the sporophyte generation, are called fronds, and they produce sporangia and sori. Singular of sori is sorus. The spores are produced in these sporangia and give rise to the gametophyte generation. So you're a lab student. How do you distinguish between these three? Well, Coledonym has dichotomous branching and a lobe sporangia. Equisetum doesn't have any leaves but has prominent nodes. And ferns look for that classic fern frond. Coledonym is the most primitive of the vascular plants. They lack leaves and do not have true roots. Instead, they have rhizoids like the bryophytes. They do have a rhizome, which is an underground stem. Their sporangia are fused together to form several lobes. The gametophyte of this plant is saprophytic in nature, meaning it survives off of decomposing material. So here's a look at Coledonym. This is actually an image from Professor Dempsey taken in Hawaii, and it's actually growing on a lava flow, on an old lava flow. Here's another look at Coledonym. You can see the lobe sporangia and the dichotomous branching here. Here you can see the fused, multi-lobed reproductive structure of Coledonym, containing spore mother cells that will produce the gametophyte generation. Equisetum has photosynthetic stems with a larger terminal strobili that contains the sporogenous tissues. They have silica in their cell walls, which makes them kind of rough to the touch. They are aquatic plants living on the sides of rivers and streams, so their proximity and the silica in the cell walls made them good for scrubbing clothes clean in the past. They are, however, poisonous for livestock. Note the small rudimentary leaves that come off of the nodes. These leaves are not photosynthetic. The stem itself photosynthesizes for this plant. Nodes in a plant occur anywhere something intersects the stem, in this case where the leaves intersect the stem. The region between the two nodes is called the internode. Here is an up-close of the strobilus, containing the sporangiophores, which produce the spores that will become the non-dominant gametophyte. Here's a cross-section you will see in lab. Be sure you can distinguish between it and the two we looked at in Lycophyta. The sporangium contains both spores and elaters. Elaters, again, are the structures that change in response to moisture levels to release the spores. When the moisture level is right, they puncture through the walls of the sporangium and the spores get released into the environment.